suggested that I talk a little bit about the graduate research that I did in Dr. Loopster's lab, and then also touch on some of the research that I've been doing in her lab. And so it's quite an array of, I worked on loblolly pines, kind of a population genetics project, and now I'm working on microbes, and it's a RNA-Seq project. So the best title I could think of was this instant messaging, where when you're studying the transcriptome, you're really getting a snapshot of what's going on in a population of cells in a population of individuals. So it's not really a full picture, but it's kind of an instant snapshot of what's going on inside the cell. So as Betsy said, I did my undergraduate in Hawaii at the University of Hawaii at Hilo. Hawaii is a really wonderful place to study biology, and especially in diversification. So when I talked to Dr. Loopster about coming to A&M, I said I wanted to study the underlying mechanisms behind phenotypic variation. And she said, oh great, I've got this project, it'll be perfect. Dr. Loopster's lab focuses on identifying regulation and function of genes involved in wood development and drought response. And it was really in her lab that I started to focus in on examining the transcripts and the transcriptome. Okay. Oh.
This way? That way? You should play with the incandescent
switches in the front.
You know the board?
The little switch, the round
Front lights? Oh, there. Okay. So we're in Texas now, and I'm in Dr. Loopster's lab, and my work there was part of the NSF-funded project called ADEPT2, and it involved UC Davis, the University of Florida, Texas A&M, and North Carolina State University. And the goals for this project were to identify naturally occurring phenotypic and genetic variation in loblolly pines, and then use association genetics approaches to correlate SNPs in these pines with variations in complex traits. And so there was, phenotyping was wood property traits, drought resistance, disease resistance, and gene expression analyses. My part here at A&M was to do the gene expression phenotyping for 88 disease and drought-related genes that I identified in the literature, and then use association genetic studies to correlate the gene expression phenotypes with almost 4,000 SNPs that were genotyped at UC Davis. The population for this project was provided by North Carolina State University, and it was a population of 354 unrelated individuals that were representing most of the range of loblolly pines. So the black border is the range, and then each tree marker is actually a county that's represented by one or more trees. I'm not going to talk about this too much. So for the gene expression phenotyping, we extracted total RNA from the main stem of every loblolly individual. We had 2 biological replicates or clones for each unique genotype, I'm going to call it that. And then we collect expression for two technical reps for every individual. We used cyber green dye for quantification, and we detected the application of a single product by dissociation curve analysis, so that the presence of a single peak. And then we also examined the amplification efficiency of every primer pair using ANOVA analysis. We found significant differences in expression among the different genotypes collected from across that range among the genotypes. And then also, as you can see in this graph, every group of 4 is a unique genotype, and every group of 2 are the replicates. So there were even differences among biological replicates, so that we're supposed to be the exact same genotype. We saw big differences in expression. The largest range of expression differences, so between the highest expressing genotype and the lowest expressing genotype, was in this gene, PTEMB4. It's a LEA protein that was first identified in a spruce, and its variation was more than 9,000-fold, so quite large. On average, we saw 20 to 30-fold range of expression differences. So our next question was, how much of this variation in expression is due to the geographical origin of these trees? And to do this, we did a principal component analysis. And we found that 30% of the variation in expression, or about 30%, could be explained by the first three principal components. And furthermore, when we examined it, we found that trees from the Atlantic coast region most often loaded into the first principal component, so that's represented with blue. You can see there's more blue in this area. Trees from the Gulf Coast region most often loaded into the second principal component, and trees from west of the Mississippi most often loaded into the third principal component. So there did seem to be a geographical trend to the variation in expression that we saw. The next part, just to remind you, we were doing the association genetics between the gene expression phenotypes and the SNP genotypes. And so association genetics, unlike traditional mapping, uses the accumulation of recombination that occurs over generations in a population of unrelated individuals, and it uses that to do fine mapping of traits. So it's very dependent on the linkage disequilibrium, on the nucleotide diversity, genome size, and availability of genomic sequence. Loblolly pine is well positioned for association genetics. They're distributed in large outcrossing populations, and this helps to maintain high levels of genetic and phenotypic variation. They can be clonally propagated. There's haploid tissue for SNP genotyping. There areas of the genome that have the linkage disequilibrium has been measured because it'll vary across the genome, show low linkage disequilibrium, and there are abundant genetic resources. So for the association study, we performed a general linear model for every SNP gene expression phenotype pair using the software Tassel. And then one of the experimental design concerns for association studies is the presence of population structure. So we accounted for this with a Q matrix, and this would hopefully prevent false positive associations. And this is all of the 384 well plates that we did for our gene expression study. There was a lot of them. So we saved them for 4 years just for that picture. So the results of our association study were we detected 101 significant associations between SNPs in 94 genes and 27 gene expression phenotypes. There were 45 SNPs in exons, and 25 of those SNPs caused an amino acid substitution. 20 SNPs did not, and then 8 SNPs in exons were in highly conserved regions. SNPs in the UTRs and in the introns, there were also a lot of those. Those SNPs could possibly be affecting translational regulatory areas or splice site recognition sites. And then there were also 10 candidate genes that we couldn't translate, and these could be possibly non-coding RNAs. So we had all of these associations, and we were trying to figure out a way to make sense of how they were linked. And there's not a lot of tools right now for loblolly, even though it's supposed to be a model organism for trees. So what we did is we found the closest homologs in Arabidopsis, not a very close ancestor or close relative, but we found the closest homologs, and then we used this web-based tool called ARNet, and it shows you all of the functional networks of genes in Arabidopsis. So as an example of kind of how we approached this, this is gene PTEMB1 we collected gene expression data on. It was a LEA protein identified in white spruce, and it was associated with these 3 genes, and they were all linked in this ARNet network here. The trick was that PTEMB1 wasn't in that network. So we kept looking in the literature and found that it was also associated with an OXIAA transcription factor, which represses the expression of auxin-responsive genes. It also requires this TPL repressor in order to fully repress. If you lose the domain for that binding, you get partial expression of auxin-responsive genes. So we went back to these 3 genes again and looked at their sequences, and we found that, lo and behold, they also have an ear domain, or the heat shock transcription factor also has an ear domain where TPL can bind. So that led us to kind of guess that hypothesize that there were two possible explanations for this. The first is that a SNP that affects the binding of the TPL co-repressor to IAA and also to PT, the heat shock transcription factor, could affect expression of downstream genes, possibly that PTEMB1. The alternative explanation is that a SNP in a gene at a nearby locus was affecting the expression of PTEMB1, and so that these were associated just because they were linked to the gene that truly had the SNP in it. So, SNP at the link locus. All of those expression experiments that we did were really interesting and everything, but they hadn't been stressed at all. The trees hadn't, because my lab mate at the time was also collecting expression on wood development genes. So for my second part of my graduate project, I decided that you got to stress the trees and measure gene expression and see, how they're behaving differently. So we designed a 2x2 experiment, and we applied 4 different treatments: pitch canker, drought, pitch canker and drought, and control, to 24 clones from the same population that were also provided by North Carolina State University. Okay, so we're doing the drought stress and the disease stress. The trees, like I said, they're from the same population. These were selected based on some data that they collected at North Carolina State University and the University of Florida to suggest that the genotypes that we got were either resistant or susceptible to drought or to disease. So that's why they were selected. And they had enough remets. So our design was, we again measured expression on the original 88 genes, and then I also selected 10 of the SNP-containing genes that we identified through the association analyses. And then, just because I felt like I needed to, I added 26 more, I guess. And we measured the main stem circumference and pre-dawn water potentials at the time of harvest. As I mentioned, carbon isotope discrimination and pitch canker lesion length, they were measured at North Carolina State and Florida. We did qPCR the same way as I did in the last experiments. So I'm going to kind of fly over these really quickly. There was significant overlap in the gene expression changes between the 2 different treatments. Only 16 genes were inversely regulated. 58 genes were expressed differently among clones within every treatment group, so all of the treatments. Drought transcription factors, drought signaling genes, drought responsive genes. Most of the genes that were involved in drought responses responded positively to all of the treatments in general. And then that PTEMB4, which is a really interesting gene to me, they sequenced the promoter region of that, and I haven't seen it yet to see what might be accounting for the big range of expression differences. But it, again, was a huge range. Now what? I didn't do anything.
This is the internet. Is this part of your presentation?
No. It's my presentation, but it's not okay. Okay. So drought transcription factors generally positively upregulated. Disease transcription factors and disease signaling genes were more mixed in their responses. Terpene biosynthetic genes generally decreased in response to drought and fusarium infections, but were increased in response to the combination treatment. And then pathogen-related proteins, on average, were induced the most. They were usually over 4-fold. Did you have a question?
Yes. So when you look at these expression size,
Yeah. Is it a significant increase or decrease?
When do you decide?
So to significantly do it, you would do like an ANOVA and then apply a false discovery rate. And I don't have that data up here on these. I'm just showing kind of the range of expression differences we saw. We performed partial correlation analyses, and we found that usually correlations between genes were stronger under a treatment. So in response to, say, drought and fusarium, it's a stronger correlation than it is in the control, and that was a typical trend that we saw. And we also looked to see if the expression of the SNP-containing gene that was originally associated with the gene expression phenotype in the association study, so they were associated, they should have been, we thought, correlated if we looked to see if that was true. And only 2 of the 10 cases were they correlated. So this suggests that it's likely that it's a SNP and a link locus that's causing that correlation rather than this actual SNP that was detected that's causing the gene expression phenotype. And then we also had some of those measurements that we took at harvest, and we had some of the data from North Carolina State and Florida. So we did a ANOVA analysis and controlled for if the phenotype was resistant or susceptible to drought or disease. And we found a few genes that were differentially regulated between resistant and susceptible genotypes. Here, NCED is a control point in the ABA biosynthetic pathway, and its expression is higher in resistant individuals than it is in the susceptible individuals in those experiments. When we did ANOVA controlling for pitch canker resistance, we found lots of terpene synthesis genes. And the surprising thing was they're expressed lower in the resistant individuals than the susceptible individuals. And at first I thought that was a little bit odd, but then when I thought about it, it makes sense. It's in the drought treatment group, and it's well known that terpenes are decreased under drought conditions. So this is probably reallocation of resources or crosstalk between signaling pathways. So in conclusion from this study, we found abundant variation in expression of disease and drought-related genes. A small amount of the variation could be accounted for by their geographical origin. Correlations between induced gene expression was generally stronger than correlations between uninduced gene expression. Using association genetics approaches, we identified 101 significant associations between SNPs in 94 candidate genes and 27 gene expression phenotypes. And once these SNPs are, once they're verified, they'll do this again in another population. Once they're verified, hopefully they'll be used in breeding programs to improve these traits. So that's the ultimate goal for this project. And then finally, that's what I said. Okay. Yeah.
Can we ask a question?
Sure.
Before you go on? So, to what extent do you think that this geographical variation is genetic, and to what extent do you think, like if you did a reciprocal transplant type experiment, to what extent do you think it was ecological?
I think that there is quite a bit of actually, it's got to do with the geographical origin. I mean, I think there's truly a genetic difference. Somebody else at UC Davis that was involved with this project has done some correlations with SNP genotypes and climate gradients, and he's found correlations between SNPs and climate. So I think that there probably, some of them are truly selected for, and some of them, might just be, natural variation that's not really just random. Yep.
And I think this is not only the hypothesis.
Right.
Hypothesis until you really do any kind
Right.
of
You have.
cross-translation.
Right. Yeah.
And it's really possible.
What's that?
It's really possible.
Well, loblolly is, you can transform loblolly, but I think that their approach to verifying these things would be to do the same experiment again and see if you got the same results, if you detected the same SNPs and the same associations. And then if you did, you would start doing crosses and things. So I think that their approach is not not the same functional approach that you could use with Arabidopsis or something like that, where you can transform easily. That's how they would verify. That's how they plan to verify anyway. Yep.
Yeah, there's a difference in good quality between your receptacle and drug.
Do I know if there's a difference between?
Wood quality.
Wood quality. Actually, I don't know. Yeah, they've collected metabolite data on this, and that they're not done with that yet. So, be good. So the second part of my talk is about the work I've been doing in the Pearson lab. And I've been using RNA-Seq to examine the effects of altering phenazine production on global transcription in two pseudomonads, Pseudomonas chlororhynchus and Pseudomonas aeruginosa. And since RNA-Seq is sort of a new, a newer approach, and maybe not everybody is familiar with it, this part of the talk is going to be pretty heavy on the technology behind RNA-Seq and the methods that I used. And I think that also a few of you in here might be interested in doing an RNA-Seq project. So if there's a step in the library preparation or anything like that that you have questions about, you can just stop me and I'll do my best to answer along the way. So first, to introduce RNA-Seq, it's also called whole transcriptome shotgun sequencing. And this is high throughput sequencing of a cDNA population to get a snapshot of the RNA transcribed from a genome of interest. So again, like I said, this is a snapshot of what's happening at one specific moment in time in one population of cells. The advantages to RNA-Seq over some of the previous transcriptome approaches like microarrays is that you can do de novo transcriptome sequencing if you don't have a genomic sequence. You can find genetic polymorphisms, and you can identify newly transcribed regions. Right now, the three most popular platforms, and this was a poll that was done pretty recently, are the two Illumina platforms and ABI solid sequencing. So even though that there are differences among the platforms as far as cost and read length and things like that, the basic steps to get through the process is the same. So I'm going to take you through those steps. We used ABI solid sequencing, and the sequencing was done at the UT Genomic Sequencing and Analysis Facility at Texas UT at Austin. So the solid sequencing workflow, the first step is preparation of whole transcriptome RNA. I've also put in here some of the quality control steps where you might think of doing them when you're making your cDNA library. And I'll tell you a few places where you really need to, because I had some problems. So you extract total RNA. The next step is to remove contaminating genomic DNA. You can do a bioanalyzer trace if you'd like to. We can also confirm, or you have to confirm, really the best way is by quantitative PCR. And then finally, an optional step here is to enrich mRNA. And this is optional. Like I said, we chose to do it thinking that it would reduce, you don't have to, you can reduce your sequencing depth. But kind of what I found was every kit was $425 for 6 reactions. I used 3 reactions for every sample. So I don't know that it was really cost effective. I would really look at that more closely if I did this again. The next step is the library construction. First you have to fragment your RNA, and we used ABI's heat fragmentation method. This method was very consistent among samples, but I found that it was different between species. So this is something that you have to specifically kind of play around with for whatever you're working with. So once you get it, it's good to go, but to figure out the length of time that you need to incubate is something you'll have to work on. And you want the length to be about 100 to 200 base pairs. Next, you hybridase and ligate RNA adapters to the ends, 5' and 3' end. And then you perform a reverse transcription, you purify the cDNA, and the last 2 steps, most library construction protocols will suggest that you size select and then you amplify. And we used Invitrogen's eGel size select system. It's very sensitive, and if we flipped these two, it worked really well. We got, you could easily see the size that you needed, and these gels have already wells in them, and you put water in it, and it runs it to the gel, and you just suck the water up, and it's good to go. It's easy. Once you have your libraries, they're complete. Next is you start preparing for the PCR and the sequencing. So you add your completed libraries into an emulsion PCR, and this includes PCR, reagents, primers, and then these little beads here. You run it through a PCR, and then once the PCR is over, you are able to enrich the beads that have extended templates here. And then you do a 3' end modification, and this allows the beads to be covalently bonded to a glass slide. And that glass slide is what you're going to sequence from. The main difference for solid sequencing versus other platforms is that most of the other sequencing platforms is polymerase driven. So you've got a polymerase that's adding the fluorescent probes. Solid sequencing is driven by ligation. So the steps for this are, in the first step, you have a sequencing primer that's bound to your template, and then you add probes, a big mixture of probes. And these probes are dye-based probes, so that means that there are two bases that have to match. So the complementary dye-based probe binds to your template, and then it fluoresces. And once the fluorescence is detected, that dye-based probe is clipped off. So then there's 5 bases remaining. So you sequence the first 2, it's clipped off, and now you've got the 6th and 7th base are open for the next dye-based probe to bind to. And then this procedure is repeated. It fluoresces, it's clipped off. And this happens for 7 cycles to extend your template. So it's about 50 base pairs. And then after 7 cycles, the whole reaction is reset. That first sequencing primer is removed, and a new sequencing primer is anneals, and it's anneals 1 base pair back. So this happens 5 times. There's 5 different sequencing primers. So in the end, every base pair on your template is sequenced twice. So this method of sequencing is very, very accurate. It's really good for SNP genotyping compared to polymerase-driven sequencing. Once your sequence comes off the pipeline, you can either assemble de novo if you don't have a genome sequence, or you align to a reference genome. And from there, you have several different uses. You can do SNP analysis or look for splice variants. We were interested in expression profiling, and then we performed differential gene expression analysis to find differences in expression among different treatment groups. So back to our original project and tell you a little bit about the biology of the organism. The 2 bacteria that we're working with, Pseudomonas chlororhynchus and Pseudomonas aeruginosa, are both fluorescent pseudomonads that produce phenazines. And phenazines are colorful, diffusible bacterial secondary metabolites. And the core structure of phenazines looks like this. And then different species of bacteria have these different modifying enzymes that add different sort of decorations, different R groups that determine the color and redox and solubility of the phenazines. So phenazines have been pretty well studied, but their real primary role in bacterial physiology is still not quite that well understood. They've been implicated as virulence factors, rhizosphere components involved in biofilm formation, fungal inhibition. They've been suggested to be signaling molecules, energy generation, redox balance, and involved in iron availability. So all kinds of range of functions. Phenazines, the biosynthesis of phenazines is under quorum sensing control, and the biosynthesis, there's a conserved set of core genes that make that core part of the phenazine structure, and then they have these modifying enzymes, like I said, that make all of the phenazines the different colors and with their different properties. So the Pearson lab, before I came, they had found a two-component regulatory system that, when mutated, uncouples phenazine production from quorum sensing. So when we were doing our RNA-Seq analyses, we were comparing these overproducing phenazine mutant strains with wild-type strains, and then we also had a strain that was lacking phenazine production. So here's a kind of just a little brief, some of our preliminary results, RNA-Seq results. We received about 6 million reads for every sample that we submitted to them, and about 60% of the read counts that we got mapped to the genome, so that's a good percentage for them. It looks like Aeruginosa mapped slightly better than Chlororaphis. The Chlororaphis genome is, the first annotation is just coming out, so it might be a matter of just genome annotation that accounts for some of that. One of the first things that we noticed was that there was a region of the genome where there was probably 20 or 30 genes that were differentially expressed in the phenazine overproducing strain and also in the strain lacking phenazine production. So it appeared to be regulated by phenazines or influenced by phenazines. And this region also has characteristics of genomic islands. It has a different GC content, and it has integrases and different transposable genes involved in like transposable elements. So for the first comparison, we were comparing wild type with RPEA, which is a strain that overexpresses phenazines. We detected 246 differentially expressed genes, and the largest majority of these were hypothetical and classified. So some of these genes would be really interesting to kind of focus on and do some knockouts or overexpression analyses. The other class that had the second most were transcriptional regulators. And also in 3084, where we had FESB knockout, so not producing any phenazines, transcriptional regulators were again the second largest class. So this suggests that maybe phenazines are involved in regulating transcription or as a signaling molecule. Oh yeah, and in this we also found 195 differentially expressed genes. In Pseudomonas aeruginosa, PAR S is again it's overexpressing phenazines, and we also had PAR R, which is the second part of the two-component regulatory system. Its differential gene expression was really similar, so I'm not showing that. But we detected 246 differentially expressed genes. Again, the hypothetical genes were the most, but there were also genes regulated in transport of small molecules and membrane proteins, which I thought were really interesting since this phenazines seem to influence biofilm formation. So we have, for every differential gene expression analysis, we have almost 200 genes that were identified. And we'd like to sort of narrow that down and focus on some genes for knockout or for overexpression. And we weren't exactly sure how to go about this. So I found an article about using these Bayesian networks and tried my hand at it. So to sort of prove the concept, I put some genes in that have been shown to be, there is work that shows how they're involved or how they're interacting. And when I put it in, it seemed like it worked pretty good. I forgot to put GAC-S in, and I should have. So GAC-A is a global transcriptional regulator. It placed that at the top of the network. It has the phenazine biosynthetic genes regulating one another, or the dotted line means that they're not sure which one is regulating the other, but they're all together. RSMY and RSMZ are together. So it seemed like it was working pretty good. So my next step was, it gets pretty complicated. But I took the genes that were found to be differentially expressed, and then I selected genes that were correlated with the phenazine gene expression, and I made a network. So here's phenazine biosynthetic genes, and then these genes are in a network with it. So I think what we'll try to do is maybe target some of these genes that are adjacent to the phenazine biosynthetic genes or above and seem to be regulating for further functional analysis. So I don't have a summary for that because it was mostly talking about RNA-Seq and how it works and stuff. But if anybody has questions, I'd be glad to take them. Sure.
With the first part, for the low-early time, is it possible to identify a tree that is resistant to canker and drought resistant for nurseries to propagate?
It would be possible to identify. I don't know if they'll ever find it though, because there's such a trade-off. Those two, drought resistance and disease resistance, there's a lot of trade-offs, I think, between the two. So I think you may find something that's drought tolerant, but then you might find out that it's really susceptible to, so their traits are not linked really. Mm-hmm.
I was just curious, you said you went with the ABI solid,
Yeah.
and we have 2 alumnus here on campus, and it seems like the library prep, the emulsion PCR stuff is a little bit more tedious with the ABI than what you have to do on the alumnus. So just can you explain why you went with
Sure.
that?
Yep.
If you were to do it more RNA-Seq, would you
Yep.
rethink what you
We did strand-specific sequencing, and I liked their library prep a lot better. They have you do they have you fragment the RNA first, and then you kinase, and you attach the adapters, and it gets rid of a few biases, I think. And I didn't have to do the emulsion PCR. They did that for me, so I just did the library preparation, and then they did it. The other reason is that UT has a lot of experience with more experience with prokaryotes than we do here. So I felt like I was in good hands because I'm new to this also.
So if you were to do it again, would you continue?
I think I would stick with it. To me, the trickiest part about ABI is sequencing is that it's the dye-base sequencing. So when you get to the data analysis end, it's a little hairier because when you try to convert from color space into base space, you can get mistakes, and so that's the trickiest part is actually the data analysis. The actual library prep and everything I thought was good, and I think that price-wise, I've heard people say that Illumina is cheaper, but I don't see that. And I don't really know how they compare either, because this way you get read counts, and Illumina you get like coverage or something, you get like 34 something. Do they give you read counts? Oh, see, when they were quoting us, they said something else. But. Anybody else?
So how about, building on that last question, how about availability of analysis tools downstream? Is there any difference?
Yeah, so for Illumina, it's a lot easier because base-based tools, I mean, you've got the Galaxy server online that's got all kinds of tools. And so once you get it aligned to the genome, you can convert from color space to base space, but that part of the process was painful for me. So Illumina has definitely got an advantage as far as free software and tools available online, and more people are familiar with it. So if you're looking for, like if you're troubleshooting and everybody is having a problem in Illumina, it's definitely online, but if there's a problem for ABI, it's not as common. So troubleshooting would be easier, I think, too.
Now, will the CLC genomic workbench, will that handle ABI?
I didn't try CLC at all, so. Mhm.
Can you elaborate the biology of this? Are we looking at the deregulated biosynthetic pathway upregulation or response of the bacteria to its own metabolite?
Sorry, I didn't get the. Are we looking for a response of the?
By looking at this transcriptional profile, are you looking for the genes that deregulated by, for example, killing the repressor protein of the biosynthetic pathway, or are we looking at the metabolic, response
To the phenazines?
bacteria to its own metabolite?
Yeah, I think we're seeing both, and I'm sure that we're seeing a lot of like genes that are just involved in bacterial physiology. That it's just like with the trees, you're just getting a reallocation of.
So like when you look at this island of you pointed one particular area, region of the genome.
I don't know if that. Yeah, I don't know if that's being regulated by RPEA or if that's being regulated by phenazines. So that's something that we would need to tease apart. So
it's not biosynthetic.
no, it's not the biosynthetic genes, but RPEA doesn't. I mean, not necessarily only regulating phenazines. So yeah.
Is there some kind of difference in fitness between the fit one and efficient?
Oh, in the bacteria? Yeah, so when you lose phenazines, you lose a lot of things like rhizosphere competence, biofilm formation. Dongping has done some swarming assays. There's differences in swarming assays. And so there's a loss of fitness. But the funny thing is we have another strain, GAC, that doesn't have phenazines, but when you put it in a mixture, it forms a better biofilm, even though there's not. So it's really kind of confusing.
So there's no room.
But in my opinion, if we did it again, I would probably go maybe 8 million read counts or something.
If there's no further questions, let's thank